Thank you very much, ladies and gentlemen. Thank you, Gerda, for inviting, and thank you, Anton. It's a pleasure to be here. In our lab, we're interested in microscopic foundation of non-invasive imaging. And this is what's illustrated in this slide in this caricature form. So here's our neuronal circuits composed of neurons of different cell types. And these neurons now drive vasodilation or vasoconstriction, and in parallel, they drive oxygen consumption. So these are microscopic processes that on the macroscopic level, we can detect with functional MRI because of their contribution to macroscopic cerebral blood flow and macroscopic oxygen metabolism. We can also manipulate neuronal populations with obviously optogenetics, chemogenetics, sensory stimulus, anything that we can manipulate them with. We can use all our electrical recordings that have been introduced nicely this morning to look at neuronal activity. We also can detect the same type of signals using non-invasive EEG or MEG. And then we can try to build models to put everything together, which has to do with neurons driving dilation and constriction, and build our non-invasive voxel, for example, if we want to understand functional MRI. So here's something that we call the roadmap for estimation of neuronal activity at this level of cell type specific populations that have been, this level has been introduced nicely already this morning. And the first thing that we do is that we just consider contributions of different populations of neurons. So this is number J of populations. And for each one of those, we are looking for this population-specific transfer function, which is then convolved with the time course of activity of that specific population. And this applies to flow, to oxygen metabolism. So this is C for CMRO2, which is cerebral metabolic rate of oxygen, and extracellular dipole moment. Then the next step that we need to worry about is that we need to translate from micro to macroscopic scale of physiological parameters, and also from this physiological parameters to non-invasive measurements. And the reason that this is not the same is because each one of the measurement comes with its own measurement theory. And then finally, everything that I was talking about from 1 to 3 has to do with the forward model. That means that how we build our non-invasive signals bottom up. But in real world, of course, we want to solve the inverse problem. So this is my number 4. And this is not something that I'm going to be talking today about. However, it's clear that this is the real world problem. But I would like to make a very simple point that if we don't have the forward model, it's very hard to talk about the inverse. So now this is the general idea that we can extract activity of cell type specific neuronal populations as long as we have this unique combination of flow, metabolism, and electrical signatures of that activity. Now, when we go from the micro to the macroscopic scale, we of course lose some of the details, but hopefully for these different types of measurements, we will be losing different types of details. And so when we combine them together, we still can recover some information, which of course is the idea of multimodal integration. And so now I'd like to show you some experimental data and a little bit of modeling results just to convince you that this maybe is feasible. Starting with the cellular and molecular mechanisms of dilation and constriction. So this is a paper that we published a few years ago, and in that paper we were looking at optogenetic activation of inhibitory neurons only, all of them, but just inhibitory ones, trying to understand what kind of vascular response we can get under those conditions. And this is the optogenetic actuator that is currently, in this particular case, is expressed in all inhibitory neurons. So those are those black bodies in there, the excitatory neurons, so these astrocytes that are labeled with sulfurdomin dye, but not with optogenetic actuator. We have our laser beam for optogenetic activation, which is about the size of a cortical column. And so here how this experiment looks like. This flash is optogenetic stimulus, and we can see here it comes. There's dilation, there's constriction, and it goes back to the baseline. So now if we look at the time course, so because this is done in vivo, we can also present sensory stimulus, and this is a response of the same vessel to the sensory stimulus, which is the baseline, dilation, and back to the baseline, and this is the optogenetic one. Here's light, and there's a dilation, there's constriction. So now, for our field, this was quite an interesting finding to start with, because we just activate inhibitory neurons, and nevertheless we have something that resembles sensory stimulus, also has this pronounced constriction. We did some other pharmacological investigation in the study, and we realized that this constriction was due to NPY positive neurons releasing NPY and acting on the vascular Y1 receptors. So since we continued this line of investigation, and we expressed this optogenetic actuator more selectively in the cells, which are NPY positive. And so here is now optogenetic stimulation of just those cells. We still get dilation and constriction, and the constriction is due to NPY, and the dilation, we believe, is due to NO. And here is a complementary data set from Celine Mateo and David Kleinfeld's lab, and they are expressing calcium indicator in the same type of neurons, or neurons with a lot of overlap with those that we're looking at. And now we can see that this is the dark one is the vessel diameter, arteriolar diameter, and the light orange one is calcium in this type of neurons. And then if you look at the cross correlation, you can see that the calcium in those neurons is actually preceding dilation, which should be the case if it's the neurons that drive dilation. So now here's something interesting about these neurons. This is just a histology figure to show how much of this green, which is presence of the optogenetic actuator, you get in the tissue when you express this actuators in all inhibitory neurons. And this is how sparse you get it when you only express it in this NPY positive neurons. And so now, nevertheless, when you do this optogenetic stimulus and you look at the dilation, this is in the first case, you can see there's some dilation and constriction. And in the second one, there's this big dilation and big constriction. So these cells are very, very vasoactive. Nevertheless, if you put a laminar electrode and you were to look at the electrical response to the same type of stimulus, like here, for the sensory stimulus, you obviously are going to see your normal type of laminar LFP. However, when you do optogenetic stimulus, there's nada. And this is not super surprising because the cells are so sparse, and also they are not aligned, so each one is oriented in a different way. What this means really is that under these conditions, you're going to get both functional MRI, but without electrophysiological signature. So the bad thing about this is that we don't get, ideally, we would like to have different types of neurons eliciting different types of signatures. So in here, we just get none. However, a good thing about this, if you do get a condition, and there's some reports in prior literature that you can see hemodynamic response, but you do not detect any electrophysiological response, this is likely to be inhibition. And this is because when you have excitatory neurons, pyramidal cells active, you usually detect very clear fields. And this is what's illustrated here. This actually was our control experiment because we needed to show that when we activate inhibitory neurons, we don't get a rebound response in pyramidal cells. These are just trials. This is multiple units, MUA and local field potential, LFPs. recorded for each one of these trials. This is for short stimulus. We only activate inhibitory neurons here, and this is for long stimulus. We can see that in the beginning, there's some MUAs because we activate these neurons. There's a little bit of LFP that doesn't continue much, but there's nothing at the end, which kind of argues against this rebound response in pyramidal cells. But this also is a good indication of how useful this classical electrophysiological measures are, which is no news for you guys, and we just talked about this extensively this morning. So I just want to spend a few minutes talking about the importance of optically transparent electrode arrays. We talked about neuropixels, but the problem with neuropixels is that it's very difficult to do optical measurements together with neuropixels because they're not transparent, and also they obstruct the axis for microscope objectives. So this is from a paper that we published a few years ago. And in this paper, we used graphene electrode arrays. This is in collaboration with Duygu. She's at UCSD, and she's the one who is designing and manufacturing this array technology. We applied this, in this case, this was done under anesthesia, and this is an array that is placed on top of the cortex. You actually cannot see the contacts because they're transparent, but you can use them to record local field potentials. So there's some local field potentials, and there is some calcium with the neuropil that you can record simultaneously using 2-photon imaging, and get some relationships which are consistent with prior literature reports. If you are interested in optical engineering, like what we are, we'll see that the presence of this array, it doesn't really change the resolution, but it does make slightly fuzzier imaging. However, what we did not do, but what can be done, is that one can use adaptive optics to correct for this effect. So now here is another type of electrode arrays that we are working with. So this is Shadi Dai. He is also at UCSD in electrical engineering, and this array on its own is not really transparent. However, the electrodes are very small, and because they're small and light as a go around corners, as soon as you're about 50 microns deep into the tissue, you actually can with 2-photon microscopy, you can image without any interference. These electrode arrays are very good in the sense that not only that they record LFPs, they also record MUAs, and this has to do with the combination of their impedance and their size. So these are very clean data because it was obtained under anesthesia, and recently we have been pushing this for recording in awake mice. And so here's an array, and this is some protective device that protects the preamplifier board and the connector. On the mouse, this is a mouse avatar. This is a real mouse wearing this device. And here's an example of data. We can look at this. We can do calcium imaging at the same time. So this is calcium indicator GRGECA. And here's an example of some calcium imaging in local field potential. And this is one single trial where you can see that we have some slow waves. So our mouse is awake, but it's spacing out. And then the stimulus comes and the mouse wakes up. And so now we get this desynchronized activity, and this is nice. So moving on to the oxygen consumption. For oxygen consumption, we're using these 2-photon visible probes that are generated by Sergei Vinogradov at UPenn. And these probes really generated a revolution in our ability to image oxygen with very high resolution. They're phosphorescence lifetime probes. So here you can see that there is, I lost my, for some reason, forgot about it. Okay, so it's a phosphorescent probe, and we have to look at the phosphorescence lifetime decay, and this decay is mapped onto oxygenation, PO2, which is partial pressure of oxygen. We can get this totally amazing maps of intravascular oxygenation. We can deliver it in tissue, and then we can look at tissue oxygenation. We helped Bruker to make it into the product, and I have no financial interest in Bruker. And so this is an example of how we make it more quantitative. We look at oxygen gradients around diving arteries and take advantage of the fact that there are no capillaries around arteries because there's so much oxygen in there that capillaries don't develop. And then we can apply this model of oxygen diffusion from single cylinder. This is something that have been used before in a muscle. We're doing this in brain. And so this is just a proof of principle that this works. We change anesthesia and we look at our profile of oxygen gradient from the artery to the tissue, and you can see that this profile in a way opens up. It becomes less deep as the anesthesia is deeper and there's less neuronal activity consuming oxygen. So from that, we can go ahead and we can calculate cerebral metabolic rate of oxygen, which is our important parameter that we input into our models. And so recently we have been doing this again in an awake mouse. And so in an awake mouse, we have to deliver the probe because it's an extrinsic probe. And this is how this type of data looks like. We have the vasculature, we know where the artery is, we follow this into the tissue. So the probe is delivered in the interstitial space, and this black are cell bodies because the probe doesn't penetrate inside. We can measure oxygen. So here is our map of the actual PO2 values, and we extract this radial gradients. So now we just need to use some math to be able to get CMRO2 out of this data. We have been recently working with Gaute and with Anders Dale at UCSD on methods to better estimate CMRO2. And this is a simulation platform that was developed by Marty Yule and Gaute Enevold's lab. So this is very pretty and also very useful, because this is something where we can, we know the ground truth. And then if we have any experimental data, such as this one, and we have these different gradients in different directions going from the artery to the periphery, they differ because vasculature is not symmetric. So there are capillaries on one side, no capillaries on the other side, and this is why these gradients behave differently. But using this type of ground truth simulation platforms, we can train our data analysis tools to try to choose directions that we can use for this type of estimation. And so with this in mind, we have been trying to address the question of CMRO2, which is the cerebral metabolic rate of oxygen consumption across cortical depth. It's a very interesting question, and it has to do with high resolution functional MRI. Unfortunately, our results show that there's no difference at all, and this is puzzling because we know that there's high mitochondrial density in layer 4, at least in the somatosensory cortex. But we think that this may be related to our mouse being awake and possibly in a different state of neuromodulation that is not entirely relaxed. And so this is why we also are interested in imaging neuromodulation. I'm gonna show you very quickly a very short piece of movie. So this is hot off the press. This is now sensor for acetylcholine, which is expressed all over the brain. This is one hemisphere of the mouse. We're looking from the top. And in here, I just want you to appreciate the dynamics. So this is, now we can look at the actual dynamic of acetylcholine release across the entire area. For those of you that are used to look at calcium, you can appreciate this is very broad, but it's still dynamic, and it's not happening everywhere at once. So there's still some cortical parcellation going on. So let me go back.
Anna, it's 10 minutes.
Yeah, I'm almost done. Skip that. And the question is, how now do we put it all together? So what we do have is that we have this vascular anatomical network model. So this is a model of the functional MRI voxel. It's based on the realistic reconstruction of the vasculature that we can populate with measurements that we obtain under the same conditions. And because this is in silico, we can also look at the diffusion and dephasing of the protons of water. We can impose different types of functional MRI sequences, and we can predict our functional MRI results. This is done in collaboration with David Boas and Sava Zakadzis. We also published lots of papers on this. Now, what is missing is connecting this to the circuit level models that we have been talking about this morning. What we currently do in this type of model is that we input our vascular dynamics, which we measure experimentally. However, in a kind of full modeling framework, you can imagine that you have your circuit model, and from the circuit model, you get all your LFP and kind of, from this calculate the non-invasive EEG and MEG. And at the same time, if you know what are the vasoactive properties of those different types of neurons, you can also predict, calculate, feed forward what are the vascular diameters, diameter changes that this type of activity is going to evoke, and you can input it to here, and you can calculate your functional MRI. And this is just in 1 single second, because we're talking about different types of futures. I want to bring your attention to this new model system that is where we can actually grow human tissue inside the mouse. So these are organoids that are grown from the stem cells. We implant them in a mouse, and so now this mouse can be imaged for a long time. We place arrays on top, and this thing on top is just to protect the array. This mouse doesn't really, it's comfortable with this enough. We can see that there is vascularization of this transplanted human organoids, and we can see that there is some kind of neuronal activity that develops with time. These are very preliminary results, but this may be useful as yet another model system. However, as you can imagine, we would need to come up with all these new modeling tools for this type of systems, because the organization is going to be very different from the organization of normal cortical tissue that we are experienced with. And so I just want to thank everybody in my lab. We used to be at UCSD. We just moved a few weeks ago. We came to Boston, so now we are at BU. And that's the end of my presentation. Thank you very much for your attention. Excellent.
Thanks, Anna. We have a question here. It's from Ben Huang. For the graphene electrode, how chronic can the probe be? Can it be implanted for months without inflammation and other long-term impacts on the tissue?
Yeah, so we didn't try for longer than a few months, but we definitely tried for a little over 2 months, and it just stays there. We can definitely record. So the issue with the graphene is that at least in its kind of current generation, we are not in a good position to pick up units or multi-unit activity. We're good in picking up fields. So this has to do with the graphene pads being large. They're about 150 to 200 microns each pad. So we cannot look at units that much, but fields are very good.
So now if we talked about talked a little bit, and we talked about it earlier, how you can model actually LFP, and if you have a circuit model, you can model LFP and EEG and also MEG. But if you want sort of to predict from a network model the fMRI signal, the bold signal, what is the best sort of proxy? I mean, there's been all this hypothesis in the past that it was like extracellular calcium or whatever, or potassium or whatever. But what is your best sort of best predictor? How should you model? How should you predict the fMRI signal based on what we compute?
So I don't know how how to predict it based on things like calcium or LFP or MUA, because it depends very much where exactly it's coming from. You know,
Right.
so when we started this, when I started this research 20 years ago, we had this very simple ideas that we can come up with some transfer functions from, let's say, MUAs or LFP and get our prediction for functional MRI. So that doesn't seem to be the case. However, if you do have some kind of knowledge of cell type population specific activity, that seems to be a common theme for today.
Yeah.
You know, everybody's interested in this. Everybody in neuronal modeling, purely neuronal modeling, is also interested in this, right?
Yeah.
This is what you guys did in that cerebral cortex paper. So if you have that level of understanding, we should be able to use this to predict functional MRI, because we know which of the cell types are vasoactive and in which way.
Yeah, exactly. So you can sort of base on the activity in one class of neural models in your circuit model, then you can sort of predict some kind of release of vasoactive.
Just a measure of spikes without knowing what cell types those spikes belong to, in my mind, are a little useless. But I think that's kind of what I heard this morning as well from other speakers. So I'm not alone feeling that way.